will not have any of those. I won't have any of those.
That's always your position.
I mean, if they do have inflation and stuff. Like a couple of cents every year.
Yeah, cost of living raise and then they give us that. And I don't even remember the last time we got one.
Yeah. It will, they factor into just doing it.
Well, I don't know, maybe I'll do that if it's too many. I just have to see how much they operate.
Keeping it up, sorry.
Well, then she left too. I don't know.
Oh, okay.
Should we wait for her? Do you guys, should we get going? Or?
Doesn't matter.
A bunch of people said they weren't coming in.
Yeah.
So, I guess we'll get going. Maybe we'll try to get 2 chapters done. Or 1.5 at least, since we're a chapter behind. So today we're going to be covering signal transduction. So, do you guys know what that is?
Signal transduction causes genotypical change with phenotypic expression.
Yeah, that's one thing.
That's Dr. Smith's little mantra.
Is it? Is that from immunology?
And hematology.
And hematology. That's right. You took immunology and hematology. Well, that's a good little mantra. But how does that work then? You learned about G proteins.
Yeah, there's lots of different ways, right?
Yes. So that's
Receptors and there's toll-like receptors.
yeah, that's more specific to immunology, I think.
We're doing immunology too.
So we'll look at just the general features of signal transduction. Specifically, we'll be looking at G protein coupled receptors. Do you know what the G stands for?
Glycoprotein?
No. GTP binding proteins.
Yeah.
Glycoprotein is a good guess, though. I would have guessed that. Then the structure and function of enzyme link receptors. You got any examples of these enzyme link receptors? I don't know if CCAM is. I think it is, you're right. The tyrosine kinases.
Okay, I remember those.
Receptor tyrosine kinases, such as like insulin receptor, things like that, or the FGF receptor, things like that. Gated ion channels, we were just discussing some gated ion channels. You have voltage gated ion channels or ligand gated ion channels. And then physiological processes using signal transduction, we'll look at some of those too. So biological role of signal transduction is, cells are going to receive signals from the outside. Since we're multicellular, like if we were unicellular or monads, they're going to have to respond directly to the environment, and that's going to require some signal transduction also. But with us, we need to coordinate all our tissues and everything together. So we have hormones is one way to do that. Neurotransmitters, how to coordinate transmission of information across neurons or something like that, right? Light, so we got to be able to respond again to environmental signals, and that includes touch and smells or pheromones. And then antigens, what you guys were talking about, those are involved with your toll-like receptors and things like that. I've never... JAK, STAT, those are all, signal transduction pathways that we'll be looking at it. So these signals, they're going to cause a change in the cell composition and function, like you were saying, genotypic changes causing phenotypic expression, right? So, and differentiation and antibody production are some of those, and that's your example, immunology. Growth and size or strength and sexual and asexual cell division are all things that can result. So receptor, what is a receptor? It's basically a membrane-bound or soluble protein or protein complex which can exert a physiological effect after binding its natural ligand. So receptors are going to bind ligands, and the receptor is what then transmits that signal. And we have G protein coupled receptors, or GTP binding protein coupled receptors. An example of that is epinephrine receptor, insulin receptor, and I screwed that up, didn't I? Not the insulin receptor. The enzyme link receptors is an insulin, or is an example of an insulin receptor or FGF receptor. Ligand-gated ion channels we look at as nicotinic acetylcholine receptors. Other membrane receptors include integrin receptors, and then you have nuclear receptors, such as your steroid receptors. And that's where your steroids are going to diffuse through your membranes, because those are lipids, right?
Yeah.
So they can diffuse through the membranes, and there's a soluble receptor that can then bind the steroid and then act on the genome in the nucleus. So 5 main features of signal transduction. The first one is it's very specific. So those receptors, just like high specificity for enzymes, you have high specificity for the ligands that are going to activate a receptor, right? And then you have an amplification of that signal. So you get binding one ligand to one receptor, and through signal transduction, you get an amplification of that signal, multifold. And another thing is that these signals can be modulated. If you have a constant presence of hormone or a ligand, that signal will be modulated to the response. So that includes like desensitization of receptors, right? And then integration. So when two signals have opposite effects on metabolic characteristics, such as the concentration on secondary messenger X or the membrane potential, the regulatory outcome results in the integrated input of both receptors. So like in neurons, you have excitatory neurons and inhibitory neurons. Have you guys heard of those before? So glutamate receptors are typically on excitatory neurons, and then you have inhibitory neurons, and both act on synapses at the dendrites, and they will modulate that action potential that's from the excitatory neurons. If you have inhibitory neurons, they'll kind of deaden that action potential, so it might not reach the threshold where you actually reach the action potential down the axon. So that's why, if you've heard of GABA, so what are those GABA potentiators?
Like what do you mean?
The GABA potentiators.
I know they do.
Yeah, the drugs.
Oh, benzos.
Yeah, the benzos, they're GABA potentiators, so they enhance the activity of GABA, which decreases neurotransmission. That's why you get, like that hypnotic effect from those.
Does it do something with action potentials too? Like
Yeah, because GABA will inhibit the action potentials, the more GABA responses you have. So they're inhibitory neurons, whereas they compete with the glutamate, and the glutamate will try to excite those, whereas if you have a lot of GABA around, it will inhibit those neurons from firing. Does that make sense?
Yes.
This isn't in the chapter at all, but just an example. So receptors bind specific ligands. Typical ligands are, you can have small ions. Ferric ion is a bacterial ferric receptor. Organic molecules such as an adrenaline or epinephrine. Polysaccharides, heparin and fibroblast growth factor include some of your ligands. And then you have peptides such as insulin. What's the opposite of insulin? Glucagon. Yeah, glucagon's another peptide. And glucagon and epinephrine have very similar physiological results, just at different targets, right? And proteins, vascular endothelial growth factor, FGF receptor. What's that going to do you know?
Feels like the
Blood vessels. It'll cause new blood vessel growth. So this is actually a target for targeting cancers. If you can block VEGF receptors, then you won't get new blood growth into the tumor, right? So some of the signal.
Examples of signal molecules?
Yeah. No problem. So the specificity, signal molecules fit the binding site on its complementary receptor, and other signals do not fit. So they're very specific, so you won't get mixed up there. And then a number of them signal through the membranes, right? So we have G protein coupled receptors is one type that we're going to look at. Receptor kinase, and these are characterized because there's a protein that binds to GTP or GDP. So then receptor tyrosine kinase activity. So these are typically, when you get ligand binding to one, they'll dimerize. Examples like the insulin receptor are already pre-dimerized. They're held together by disulfide bonds, but a number of them aren't dimerized, but you get dimerization after ligand binding, which causes activity or kinase activity in the intracellular part of that molecule. Receptor guanylyl cyclases. So ligand binding triggers a guanylyl cyclase activity to occur, which then cyclic GMP is then a secondary messenger inside the cell. And then gated ion receptors, voltage gated or ligand gated ion receptors. And then we have adhesion receptors, which are connected to our cytoskeleton, and they can cause rearrangements in the cytoskeleton and motility based on what kind of extracellular matrix materials it's binding to in the extracellular matrix. And then we have, a lot of these are going to cause a kinase cascade. So can somebody tell me what a kinase is?
Phosphorylase?
Yeah, so it's not a phosphorylase. It's an enzyme that adds phosphate groups. It phosphorylates things. Phosphorylases.
Take away phosphates.
No, those are phosphatases. So, but we will see a phosphorylase when we talk about glycogen metabolism, because that's actually a phosphorylase where you add a phosphate rather than a water to split that bond between the sugar and the glycogen polymer.
Metabolism.
So, huh?
That's in the metabolism one?
Yeah.
I remember that kind of thing.
Yeah.
That's long.
Yeah. So that's a phosphorylase where it adds a phosphate across a bond, replaces, a bond between two. Typically it's a phosphodiester bond again between a sugar and another phosphate, and you replace that one phosphate that's bound with a free phosphate. That's a phosphorylase. But we're digressing into things we don't need to know right now. So, and then I forgot to mention that nuclear receptor. Again, nuclear receptors are hormone binding. They're typically steroid hormones. And they don't need a membrane receptor because they can transverse the membrane because they're lipids themselves. Make it to the nucleus, and when they bind to the receptor in the nucleus, they'll alter gene expression patterns. So we'll first start off talking about G protein coupled signaling. So there are typically 7 alpha helixes that span the membrane. And then they're a heterotrimeric, where you have the receptor, and then you have this alpha, beta, and gamma portion of the protein that associates with the transmembrane domain. And the alpha, beta, gamma portion binds to GTP or GDP. And the GP proteins will mediate signal transduction from G protein coupled receptors to other target proteins. And one of the examples is this. We have this G protein coupled receptor. What happens when you have binding of a receptor to a ligand is it's going to activate cyclic AMP as a common secondary messenger of G protein coupled reception. You get ligand binding, which causes GDP to be displaced with GTP. Once GTP is bound to the alpha subunit, it will cause it to diffuse away from the beta and gamma subunits, and the alpha subunit will diffuse to a secondary messenger system. In this case, it's adenylene cyclase, which will take ATP and produce cyclic AMP. And cyclic AMP is that secondary messenger, which will then go on to do other things, such as activate protein kinase A, or CAMP activated kinase, right? Is that what they say in the book?
What was that? Half of that ligand?
So you
What is this?
So the ligand binds, the alpha subunit replaces GDP with GTP, which then diffuses away from the membrane-bound receptor and activates, when it's bound to adenylyl cyclase, it'll activate enzyme activity to convert ATP to cyclic AMP.
I understand. I'm wondering specifically what channel or what mechanism is this?
These are the
Is it
G
specific to a certain receptor?
Yeah, any of the G protein coupled receptors.
Okay, that's what I was trying to remember.
Yeah, so yeah, remember G proteins because it's the GTP here. And that's where it gets its name because it'll substitute GDP to GTP. And then it activates some secondary messenger signaling after that. And when you have cyclic AMP around, what it's going to do is it's going to interact with the regulatory units on protein kinase A. So protein kinase A exists with 4 subunits, 2 regulatory subunits, and 2 catalytic subunits. When the regulatory subunits are bound to the catalytic subunits, it has no catalytic activity. So it's a protein kinase. So anything where you see kinase means it adds phosphate groups. Protein kinases add phosphate groups to other proteins. So protein kinase A.
This is the one in between the extracellular membrane and the nucleus?
Yeah, this is in between the extracellular membrane and the nucleus. So cyclic AMP is going to bind to the regulatory subunits on protein kinase A. And when it binds there, they dissociate from the catalytic subunits, leaving the catalytic subunits available to phosphorylate proteins. And that's how that message will then get added. One example that I use here is that it's CREB, which is cyclic AMP response element binding protein, I think is what the CREB stands for. So when CREB is phosphorylated, it'll then bind to that CRE, which is the cyclic AMP response element on the DNA, and you'll alter transcription. Does that make sense? Sort of? Yeah. So this is just a prototypical G protein, RAS, which shows the G protein, or the GTP binding domain, along with magnesium. So, and then the next figure is a number of different G protein coupled receptors examples. They're crystal structures. So we have beta-2 adrenergic receptors, morphine analog ligand binding site, or morphine U-opioid receptor, and the histamine H1 receptor. And then this figure here is just them all merged together, showing you the similarities in the actual receptor transmembrane domains. And then here's your alpha subunit, beta subunit, and then your gamma subunit. And if you notice here, they're all S's. You guys have any idea what those S's stand for? Stimulatory. So you have stimulatory G proteins, you can also have inhibitory G proteins.
So the ones below the membrane are that would be the alpha?
Yeah, so this is the alpha subunit. If you get ligand binding, their GDP here is going to be kicked off, and GTP will then bind. And with GTP bound, the alpha subunit no longer has an affinity for the receptor and the beta and gamma subunits, and it'll diffuse to its secondary messaging unit.
Because there are medications that are alpha 2 adrenergic receptors, and that's why I was asking. I see the beta 2 adrenergic receptor.
Yeah, they're just different receptors.
I'm just wondering if they would be on the inside of the cell.
I'm not sure about that.
It's not important.
But these are what blocks, these are the beta blockers.
Okay.
Act on these. Are you familiar with those? So, huh? Yeah. So, and one of them that binds to these is epinephrine, right? So we release epinephrine when we're scared or excited, right? So that's basically mediates our fight or flight response. So it's going to tend to mobilize energy. So it has a lot of, if you release epinephrine, you're going to start to mobilize glycogen and start kicking out sugar into your blood, basically. So binding of the receptor in the muscle or liver cells induces breakdown of glycogen. And then binding of the receptors in the adipose tissue will induce lipid hydrolysis, and that'll start kicking out free fatty acids and ketone bodies. So binding to these receptors in the heart cells will increase the heart rate also. And it's very specific, so you can see epinephrine structure. You have isoproterenol is an agonist that adds to that, and then you have what was it?
Propranolol.
Propranolol. Basically blocks it. So, antagonists. And this is how what you get. This is again the figure from your book. You have epinephrine binding to the receptor. When you get binding, GDP is going to pop off the stimulatory alpha subunit, and GTP will associate with it. Once GTP is associated to it, it activates adenylyl cyclase, which will produce cyclic AMP. Cyclic AMP activates protein kinase A's, like we just saw, and that will phosphorylate cellular proteins by protein kinase A, causing a cellular response to epinephrine. And that includes proteins involved in the glycolysis and gluconeogenesis pathway. It blocks gluconeogenesis, activates glycolysis, and it activates glycogenolysis, or the breakdown of glycogen, okay? But these subunits, they can't just stay active forever, right? So there's GTP ACE activity that will eventually convert this GTP into a GDP to turn this off again. So eventually this will get shut off. And likewise, cyclic AMP can't hang around forever inside the cell because there's so many things that responds to it. Just like other secondary messengers like calcium has super low concentrations in the majority of the cytosol, or in the cytosol. It's really low because that acts as a secondary messenger also for many. You have to turn this off, and their cyclic AMP is degraded by the reversing the activation of protein kinase A. So, it's degraded by cyclic nucleotide phosphodiesterases, which ends up reversing that.
Wait, where is PKA in here?
So PKA, they don't show it to you like I just did
Yeah.
with the 4 subunits. They just have an arrow pointing saying it activates it. So, and this is the overall synthesis. You get a cyclic AMP. So you have ATP, and then this catalyzes. This hydroxy group will attack the phosphate, releasing pyrophosphate, which is right here. And then you have cyclic AMP, and then cyclic nucleoside phosphodiesterase replaces that with water on the 3' hydroxy, inactivating the cyclic AMP, releasing AMP. So signal amplification in the epinephrine cascade. So activation of a few G protein coupled receptors will lead to the activation of a few adenylyl cyclase enzymes, and they'll produce quite a few cyclic AMP molecules, and that'll activate several protein kinase A enzymes, and then these will activate thousands of glycogen degrading enzymes in the liver tissue. And at the end, you have tens of thousands of glucose molecules that are spit out of the liver into the blood. So, and why would you need that?
To respond to that.
Yeah, some fear, or you know, if you're trying to fight for your life, you need a lot of energy there. Yeah. So, and this is just a figure showing you how that amplifies. So you get 10 molecules, activate cyclic AMP, which results in 200 molecules of cyclic AMP, which will then have 100 molecules of active protein kinase A, which will activate 1,000 molecules of phosphorylase B. Remember how you were talking about phosphorylase?
Yes.
This is phosphorylase B, which is phosphorylase B kinase. So this is the enzyme that actually activates phosphorylase A. So this is another protein kinase that'll phosphorylate phosphorylase B, converting it to phosphorylase A, activating the glycogenolysis, or glycogen breakdown. And that glycogen gets broken down, you add that phosphate between that phosphodiester bond, and so you end up with glucose 6 or 1-phosphate, I forget which one.
Glucose 1-phosphate.
Yeah. Oh. Right there. I just need to read my slides to learn. And then you end up with like 100,000 molecules from that. So self-inactivation of G protein signaling. So epinephrine is meant to be a short-acting signal, right? So the organism must stop glucose synthesis if there's no more need to fight or flee. If you have like this super high glucose level, eventually if you just constitutively activate the epinephrine signal. Downregulation of cyclic AMP is going to occur by hydrolysis of the GTP on the alpha subunit of the G protein. And that's that alpha subunit has that GTPase activity built into it. So eventually, the GTP is going to convert to GDP, and the alpha subunit will dissociate from adenylyl cyclase and then back to a G protein. So that's one of the methods how you inactivate it. The other inactivation type of method is desensitization of the receptor itself. And what happens there, you have active receptor turned on, you're binding epinephrine to the beta adrenergic receptor, triggers dissociation of the G subunit. You get the GSBY will recruit BARC, an enzyme BARC, beta adrenergic receptor kinase to the membrane. That's what the BARC stands for, beta adrenergic receptor kinase, which will phosphorylate it. And after it's phosphorylated, BARR will bind to that phosphorylated group and cause endocytosis of the receptor itself. And then eventually those will get recycled and moved back towards the plasma membrane when it's necessary. So you actually remove receptor molecules away from the membrane so they can't respond to the epinephrine that's free in there. Another way to kill the signal, or you desensitize that cell to the free epinephrine outside of the cell, right? So cyclic AMP is a common secondary messenger. I'm sure you guys heard about it a lot in immunology. So a large number of G protein coupled receptors mediate their effects through cyclic AMP, and both activating and inhibiting cyclic AMP synthesis. And the human genome encodes about 1,000 G protein coupled receptors with ligands such as hormones, growth factors, and neurotransmitters. And there's hundreds of different G protein coupled receptors responsible for similar processes. Taste and smell, and even the sense of light is G protein coupled receptor also. So ligands for many of the G protein coupled receptors have yet to be identified. And this adenylyl cyclase, there's these proteins that can kind of coordinate and house everything to respond from one receptor close nearby, so you'll get local action rather than having the cyclic AMP here go and activate other kinases far away. It's held close to the protein kinase A, and if you see here, there's the two regulatory domains and the catalytic domain. Cyclic AMP binds the regulatory domain, releasing the catalytic domain to go hit its target proteins, which are all kept nearby with its, what do they call it? Anchor protein, right? This anchor protein. This PP2A, anybody know what that stands for?
Phosphorus, diadenine.
Protein phosphatase 2A. So protein phosphatase removes phosphate groups. As you can see here, the target protein removes that phosphate group. So that's a way to stop that signal. So PP2A is a good regulated phosphatase there also. One near to my heart. So, and then this is an example, this table in your book showing you the sequence of phosphorylation. So these are serine sites that get phosphorylated. Why is serine good for being phosphorylated?
Serine has a hydroxyl?
Yeah, serine has a hydroxyl. So it gets phosphorylated on the serine. And all of these have specific serines that end up being phosphorylated. And then some of the signals that use cyclic AMP as a second messenger include corticotropin, or ACTH, corticotropin releasing hormone, dopamine, epinephrine, follicle stimulating hormone, glucagon, and et cetera. I'll use cyclic AMP. So basically what we've talked about here is just the stimulatory G proteins. You also have inhibitory or GI alpha subunits. So the GI are inhibitory G proteins. You have GQ, which cause a response. They activate phospholipase C, or PI phospholipase C is phosphoinidazole phospholipase C, which breaks apart that phospholipid into its IP3 head group and DAG. Do you know what DAG stands for? So if you remove the head group on a phospholipid, what do you have left?
It's a long chain.
It's two long chains, right? Diacylglycerol. Okay, so that's what the DAG stands for, diacylglycerol. This is anidazole triphosphate. And that's both of these they're both secondary messengers in this G protein cascade here. And then you have your GO, which actually the alpha subunit goes and opens up an ion channel. And that influx of calcium or whatever ion ends up being the secondary messenger for that. So, and this is example of the GQ, right? So here we have a hormone, binds to its specific receptor, GDP gets knocked off on the GQS alpha subunit. GTP comes in, which activates phospholipase C, which cleaves PIP2 into IP3 and diacylglycerol. IP3 can diffuse through the cell, open up a calcium channel, allowing calcium influx into the cytosol. And at the same time, diacylglycerol binds to protein kinase C and activates it along with the calcium. And you get protein kinase C, which is then again is another enzyme that phosphorylates proteins. It'll phosphorylate its respective targets, and you get the signal transduction again into the nucleus. So, why did I have that link there? Let's see. Oh, this is a. Where is it? I saw it. Ah, there. No, not all the way. Is it playing? This is weird. I think this screen is above mine.
Extracellular signals produce specific responses in target cells through the action of intracellular second messengers. Here, we focus on 3 second messengers, IP3, DAG, and calcium, all involved in the inositol lipid signaling pathway. A hormone receptor signal on the cell surface leads to the activation of phospholipase C, PLC. Activated PLC hydrolyzes the inositol phospholipid PIP2 into DAG and IP3. DAG remains in the membrane, while IP3 diffuses through the cytosol to the ER surface. At the ER, IP3 molecules bind an IP3-sensitive calcium channel. This induces the channel to open, allowing release of stored calcium ions into the cytosol. Depletion of the intracellular calcium store activates store-operated calcium channels, SOCs, in the plasma membrane. The activated SOC opens, and extracellular calcium enters the cytosol. The transient increase in cytosolic calcium levels mediates various cellular responses, including the binding of protein kinase C, PKC, to the plasma membrane. Back at the plasma membrane, membrane-associated PKC is activated by DAG. Activated PKC phosphorylates multiple targets, resulting in a varied array of cellular responses.
That's it, right?
I thought you said there's not a lot of calcium inside of the cell.
There's not. That's why you get an influx of calcium, it triggers all these types of responses. So the intracellular concentrations are kept really low, which is one of the interesting things. When you get a response with calcium, you boost the concentration of calcium, but there's mechanisms in the cell to clear the calcium. There's pumps that'll burn ATP and clear that concentration of calcium, and they store it. There's storage of calcium in cells and organelles. Just like in your muscle cells, the sarcoplasmic reticulum is where the calcium is stored. That gets released based on an action potential from a nerve signal. Voltage-gated calcium channels open up in the sarcoplasmic reticulum, which trigger muscle contraction. And then that calcium gets cleared eventually. Otherwise, you would have, you know, what's that disease called?
Atrophy?
Huh? Not atrophy. Lockjaw.
Yeah.
Tetanus, yeah. You'd have tetania, right? Constant, where you would just be totally all.
So you said they're stored, but are they stored near the ER or?
Mm-hmm.
Calcium's also stored in your bones.
Yeah, but it's not active in that form. So you have higher concentrations outside of the cell and really low concentrations in the cytosol. So when you open either an a membrane channel or an intracellular channel, that calcium is going to flow into the cytosol and be able to act on all the receptors for the calcium, which is the next slide, if I figure out how to hide this again. There. So these calcium responding proteins, and one of the most famous one is this calmodulin, and they all have this similar type topography where you have calmodulin has a long central helix, it's a soluble molecule, and then it has like two hands that will actually grab to the calcium and give it structure. It doesn't really have this structure unless the calcium is bound, and when the calcium is bound giving it structure, it can bind to its prospective calcium or calmodulin binding sites on other proteins, right? And like you were saying, the calcium goes into the cytosol, but all those proteins actively pump calcium out. So when you have a signal introducing calcium, you'll have like these wave type fluctuations of the calcium signal. And they've actually made these cool mice where they genetically engineered them they made a fusion protein of calmodulin along with GFP. You guys know what GFP is? Green fluorescent protein. So it's a protein that fluoresces green. And they've included this calmodulin protein with it. They inserted it inside the GFP, so only when there's calcium around will that protein fluoresce green. Otherwise, it won't have the right structure to fluoresce. And then they've made transgenic mice, and you can actually, let's see. You can actually, like, they've actually put a window into the skull of a mouse, and they hit it with live imaging, fluorescent imaging. You can see the fluctuations of the calcium signals, all those neurons that light up with the fluorescent signal are actually, where is this?
That's horrible. I know it's like a mouse.
So you can actually see the neurons as they fire, like that one, boom, boom, and they kind of come in waves, that calcium signal.
So what is calmodulin?
Calmodulin is a protein that binds calcium, and then it acts on other proteins. When it's bound, let me get rid of this. When it's bound, it has this structure, and it can go on and bind other proteins. And when it binds the other proteins, it's going to either activate them or inactivate them based on what the signal is.
It doesn't have to be a specific one. It just kind of activates.
There are specific proteins that it'll react to.
Okay.
So like this is the example. These are all proteins regulated by calmodulin, or regulated by calcium and calmodulin. So. So, some of the bacterial toxins target these G protein, or they don't target them, I guess they do, target G protein coupled receptors or G protein signaling. And the result is that it causes the disease. So basically, cholera toxin and pertussis toxin do it this way. They adenylate the alpha subunit on their protein. So you end up with this adenylation group, which either inactivates it or inactivates that activity. So one of the examples here is cholera toxin. You get ribosylation on the alpha subunit, which blocks the GTPase activity. So you have this GTP stimulated alpha subunit that makes cyclic AMP, and you can't turn it off because the GTPase activity has been blocked by the the ribosylation of that arginine. So in this case, it's like constantly turned on. You can't get rid of the GTP. So you make a ton of cyclic AMP, which causes chlorine, water, and bicarb to be secreted into your intestines, and that's what causes the diarrhea of cholera. So.
I'm saying that's part of what you get the hydrate.
Yeah. Likewise, pertussis toxin ribosylates an alpha inhibitory domain. So this is an inhibitory G protein, which blocks the inhibitory domain and increases adenyl cyclase again. Increased cyclic AMP results in that histamine reaction. So the next section of signal transduction we're going to look at is these enzyme link membrane receptors, and these they basically consist of an extracellular ligand binding domain and an intracellular catalytic domain. So there's two domains. There's typically just one alpha helix spanning the membrane connecting the two. The most common are these tyrosine kinase activities. So what do you think tyrosine kinases are going to do?
Add tyrosine?
Not tyrosines. Add phosphates to tyrosine residues, right. So they phosphorylate tyrosine, so you have these phosphatyrosine residues. So this adds a phosphate. The first thing that happens, you get ligand binding, you get autophosphorylation on the enzyme itself, and that adds phosphate groups to itself, and which leads to a conformational change, allowing it to bind other proteins, and it'll phosphorylate specific target proteins after that. And then it adds a phosphate group to tyrosine and specific target proteins when it binds and interacts with those. Some catalytic domains have guanylyl cyclase activity rather than tyrosine kinase activity. So what do you think guanylyl cyclase activity will do? Huh? Yeah, it'll convert GTP into cyclic GMP, which is very similar to cyclic AMP, right? But instead of the A, you have the G. So it's like a different handle. So it'll be recognized by different things.
Wait, okay, so PKA is not an enzyme-linked membrane receptor, it's the G?
Yeah, protein kinase A is not an enzyme-linked membrane receptor. It's not a membrane receptor at all. It's one of the downstream signal cascade kinases. It's one of the first ones. So cyclic AMP binds to the catalytic, the regulatory domains of protein kinase A, releasing the catalytic domain to go phosphorylate proteins. And then they'll go phosphorylate other proteins and so forth. And you have this cascade. Yeah. And this is basically the structure, what we see with the receptor tyrosine kinases. So the insulin receptor, it's a special case because typically when you get ligand binding, it's going to dimerize before you get that autophosphorylation. Insulin receptors already pre-dimerized, held together by disulfide bonds. But for example, the VEGF receptor will have to dimerize, or the platelet-derived growth factor receptor will have to dimerize. The endothelial growth factor receptor, and TRAC-A, you guys know what TRAC-A is? I didn't. So it's the receptor for nerve growth factor, and then FGF receptor, or fibroblast growth factor receptor. And it just shows the different subunits here. They have very similar like modules that are repeated over and over again. Such IG-like domain means that the structure here is similar to an IG, an immunoglobulin-like domain. You have leucine-rich domains, cysteine-rich domains, or aspartate-glucine-rich domains, and that's just how they characterize the receptor themselves. And then, of course, tyrosine kinase domains are always found intracellularly. So, and this shows you here a little cartoon about how this works. So first you get two inactive monomers that contain tyrosine residues in the tyrosine kinase activity domains. Ligand binding leads to dimer formation, and that dimer formation will then lead to autophosphorylation, causing these tyrosine residues to be phosphorylated. Once those phosphorylated, you get phosphorylation occurs. That activates binding to other, proteins that'll bind to the phosphorylated tyrosine kinase. They get phosphorylated, and that transduces signals and ultimately results in cellular responses. So. And the one example we look at here is the insulin receptor. So it's the hormone for glucose uptake and metabolism. It's a peptide hormone. We all know that. There's like two pieces. It's made as a pre-pro insulin. A little piece gets cleaved off, and then two other pieces are cleaved off to make the active form of the hormone, held together by disulfide bonds. It's produced and released from the pancreas in response to nutrients such as glucose, and that's what helps you keep your blood glucose low. As blood glucose gets high, you release insulin, and that triggers your cells to put glucose transporters at the membrane that transport glucose into muscle cells and liver cells and such. So insulin reaches target cells such as the liver, muscle, or fat via the bloodstream. Binding of insulin to the insulin receptor initiates a cascade of events that leads to increased glucose uptake and metabolism, and the inability to make or sense insulin is basically diabetes. And actually they call Alzheimer's, they refer to it might be like the type 3 diabetes, where it's actually insulin resistance in the brain, is one of the theories behind that. So, and this is the figure from your book that shows you get activation of the insulin receptor, and that's going to end up signaling glucose transporters stored within the cells and vesicles. So you end up with these are stored in the cell. Insulin binding causes these to fuse with the plasma membrane. When they're fused with the plasma membrane, glucose can then be transported easily across the membrane down its concentration gradient, and then they'll be endocytosed again, desensitized when you no longer have all that sugar in the blood or outside the cell. It gets endocytosed and then recycled back into the larger endosomes, recycled back into small vesicles from the endoplasmic reticulum, and then it'll do it all over and over again in response to insulin, hopefully. So insulin binding to the extracellular domain of the receptor activates the catalytic domain inside the cell. So once you get insulin binding, it activates these sites to autophosphorylate each other. And receptor autophosphorylation allows the binding of and phosphorylation of insulin receptor, I forget what IRS1, insulin response, I forget what it is. I used to know that because I studied it. IRS1 then will be phosphorylated, and that activates a number of other things downstream from that'll ultimately result in those sugar transport molecules at the membranes. So indirect interactions between phosphorylated IRS with protein RAS initiates protein phosphorylations, and then ERK, one of the phosphorylated protein kinases, enters the nucleus, and a transcription factor, ELK1, becomes phosphorylated, and it'll stimulate the expression of specific genes such as the GLUT4, which is a glucose transporter. So ERK phosphorylates ELK first. It's RAS phosphorylates ERK, which then phosphorylates ELK, which produces GLUT4. So you have these crazy cascades. I think that's shown in this figure. So insulin IRS gets phosphorylated, binds to GRUB and SOS, activating RAS. When it's bound to RAF1, phosphorylates MEK. MEK phosphorylates ERK. ERK phosphorylates ELK. Ow. It's a cramp in my hand. And then that will result in gene transcription, and then that transcription results in GLUT4 receptors being produced. Another tyrosine kinase is the JAK-STAT signaling system. So JAK, again, is a protein kinase, and STATs are signal transducers and activators of transcription. And so you have like erythropoietin activates autophosphorylation on the JAK-STAT. STAT will then be phosphorylated. The phosphorylation of STAT, then you form the STAT complex. It can diffuse into the membrane, and I got a better animation than that one. And this example is with interferon receptors. So interferons will activate JAK. JAK will then phosphorylate, and they'll form the STAT complex, phosphorylates P91, P84, and P111 of the STAT complex. Those subunits come together, which will then all diffuse into the nucleus along with the P48 subunit. The P48 subunit actually binds to the DNA, and when they're all together on the DNA, that's when you get transcription at that interferon response element. So you also have crosstalk between tyrosine kinase receptors and a G protein coupled receptor. So in this example, we're looking at beta adrenergic receptors along with the insulin receptors will crosstalk with each other. So insulin, tyrosine kinase activity will phosphorylate some of the tyrosines on that tail on the beta adrenergic receptor, causing internalization of that beta adrenergic receptor. Because if your glucose is so high, if you remember, epinephrine is telling that cell to break down glycogen. If it's a liver cell, break down glycogen and kick sugar into the blood. But if your sugar is high enough, you don't want to do that while you're telling cells to pull sugar out of the blood. It's telling the same cell to pull sugar out of the blood. So it'll phosphorylate the beta adrenergic receptor, causing it to be internalized, and basically taking it out of the system so it's not being activated anymore. Does that make sense? Guanylyl cyclases, and that's another receptor-bound enzyme, and there's also soluble guanylyl cyclases. If you remember, guanylyl cyclases take GTP and make cyclic GMP as that secondary messenger. So membrane-bound guanylyl cyclases are the other group of single transmembrane segment receptors, besides receptor or tyrosine kinases. And peptide hormones activate the membrane form, whereas separate and resac for mammalian oval. So there's an example of those. Activation may also involve the oligomerization of receptors, just like receptor tyrosine kinases. Then your soluble guanylyl cyclases, they're your receptors for nitric oxide. Are you familiar with nitric oxide? Huh? That's nitrous oxide. But, yeah, I think it is, or is it NO? It's just NO. So it's a free radical. It's like a really short-term type molecule. And in fact, you do probably give it to people in the pharmacy.
Do we?
No, I don't think so.
Yeah.
Maybe that's
Not NO, but a prodrug for that. Nitroglycerin.
Oh, okay. Yeah.
So what does that do?
Dilates blood vessels.
That's the purpose of NO. So it dilates blood vessels. It causes smooth muscle response. So it's a free radical that acts as a neurotransmitter and a second messenger. And it relaxes smooth muscle. So it also stimulates macrophages to kill tumor cells and bacteria. If you learned that in immuno yet? Yeah.
But he didn't have a lot to say about it because I guess I don't really understand too much about it.
Yeah.
Yeah.
But it's a soluble receptor. So NO binds to a guanylyl cyclase receptor, and this guanylyl cyclase has a heme group that'll actually be able to bind the nitric oxide, stimulating that GC activity 50-fold. So it starts kicking out GTP when it binds to that.
I mean, it's been like a year since I took immunology, but I remember that was like something that you couldn't reliably use for diagnosis.
Yeah.
Nitric oxide.
Yeah, because it's such a reactive molecule with a short half-life.
Yeah.
You're not going to be able to see that reliably, right? So catalytic domains basically of these guanylyl cyclases, they convert GTP to cyclic GMP. And it works through the activation of protein kinase G, basically. So a lot of these signaling molecules that we see, the secondary messengers, have all these different type of adapters. And they're all like, you can basically, each piece has a different, similar structure to, it's similar to other molecules. So this is for RAS signaling. It has SH2, which has got a tyrosine phosphate group on it, SH3, proline rich, SH2 again, the pH is phosphoinidazole triphosphate responding domain, and then C2 is phospholipids, calcium dependent, and then it has this GTPase activating RAS gap domain. So they all share like similar structures, and they're just like put together with different orders, so they'll have different functions basically. So the next section we're looking at is the gated ion channels. And that basically regulates the transport of ions across the cell membranes, and it will respond to changes in membrane potentials. What kind of ion channels are those? Huh? We talked about them earlier. So there's two types of ion channels.
The action gated.
Not action, but it's voltage gated.
Yeah.
So membrane potential is basically voltage, right? Potential difference is a voltage. So membrane or voltage gated ion channels, and then we have ligand gated ion channels. There's many important roles in the nervous system. We have voltage gated sodium channels, nicotinic acetylcholine receptors, ionotropic glutamate receptors, and then gamma aminobutyric acid receptors. So this is your GABA. And remember what I was saying earlier, this is stimulatory, which is counteracted by the inhibitory neurons. And they kind of work against each other, and they modulate the signals that you get from receptors like acetylcholine or dopamine, serotonin. They'll modulate that activity. So membranes, they're electrically polarized, right? So inside of the cell compared to out is about negative 50 to 70 millivolts. The membrane potential is largely due to this sodium-potassium ATPase, which pumps 3 sodiums out and allows 2 potassiums to come in, which leaves you with a net -1 charge inside. So the flow of ionic species across the membrane depends on its concentration gradient and overall electrical potential. So that electrical potential is going to drive the sodium to want to come in. The potassium, the concentration gradient will drive that potassium out, right? Because you should have low potassium outside of the cell, high sodium outside of the cell, and high potassium in, low sodium in. Does that make sense? But because you have more negative in, that's going to kind of impede this potassium flowing out also. Does that make sense? Because it's both due to its these ions will want to flow down their concentration gradient, and also they're going to flow based on their charge. They're going to want to go where the charge is too. So if there's more negative in, that's going to cause a net influx of positive ions. And this is the figure from your book showing you the potential difference in these ATP aces burn ATP and use that energy to cause that electrochemical gradient across the membranes. So voltage-gated and ligand-gated ion channels and nerve signals. So nerve signals within nerves propagate as electrical impulses, and those are action potentials. And this propagation of the impulse involves the opening of these voltage-gated sodium channels. So once you get a little bit of a change in that membrane potential, that can cause that gate on the voltage-gated ion channel to open, causing an increased influx of sodium, which will propagate that down the cell as it leads to the axon, and then all the way down the axon. That's where you get that action potential, if you have that threshold voltage met at the beginning. So you have an opening of voltage-gated calcium channels at the end of the axon, triggers the release of neurotransmitter acetylcholine, and then that'll bind to the receptors on the neighboring neuron next to, in the synapse. And if enough acetylcholine is released, it'll cause another action potential in the next neuron. So acetylcholine then opens the ligand-gated ion channel on the receiving cell. And if you have enough acetylcholine around, and if you have, glutamate neurons exciting that cell even more, you can get an action potential. Or if you have GABAergic neurons, will kind of inhibit that potential difference, in the dendrites before you get the action potential down the axon. So ion channels and nerve signaling, and this is the figure. So you have depolarization as it flows down the axon, opens voltage-gated sodium channels. As it gets near the synapse. So it gets near the synapse, it causes voltage-gated calcium channels to open, which will cause vesicle to fuse with the membrane at the synapse, releasing neurotransmitter, causing that cycle to go on again if this is a strong enough signal in the dendrite of the next neurons.
So this relates to the nicotinic acetylcholine, so how does that really involve this? It's just like...
Yeah, so the nicotinic acetylcholine receptors, so acetylcholine is the ligand. Nicotine it's not the natural ligand, but it'll I can't remember words.
Same word that we would use for epinephrine.
Yes.
Yeah.
It's an agonist.
What?
It's an agonist. Nicotine is a ligand agonist, so it'll bind the same site as the acetylcholine. And in fact, there's two different types of acetylcholine receptors, because acetylcholine has a lot of free movement around that molecule. You have nicotinic receptors, and you have muscarinic receptors, and it's all based. And if you look at the two structures, nicotine won't activate muscarinic acetylcholine receptors, and vice versa. And it's the way that acetylcholine, the atoms on it will spin, whereas the nicotine is fixed in one shape, and so is the muscarine. Nicotine comes from tobacco. Muscarine comes from those mushrooms, those red cap poisonous mushrooms. You know what I'm talking about? So two different types of acetylcholine receptors. So acetylcholine is the natural ligand for acetylcholine receptors. Nicotinic acetylcholine receptors you find a lot in the nervous system, like associated with memory and consciousness, and also muscle, movement.
Oh, so it's something that's naturally occurring in the body. It's not brought on by...
Yeah, acetylcholine is actually built. In fact, remember phosphatidylcholine? That choline molecule is part of the head group on lipids. So acetylcholine is that choline with an acetyl group and an ester bond. And that's basically a structure of acetylcholine. And then the nicotinic acetylcholine receptors will respond to acetylcholine and nicotine. Muscarinic acetylcholine receptors will respond to acetylcholine and I can't say it. Not muscadine grapes, but anyway, and they're found, those different receptors are found in different populations of neurons and different pathways. So you get release of acetylcholine, which may cause an action potential. So this is coming from an axon. This would be a dendrite. You propagate depolarization through the dendrites. If it's strong enough, makes it through the cell body to the axon. If it's over its threshold, it'll cause an action potential down that next axon to the next nerve. So, and that's heavily modulated with, excitatory neurons that modulate the effects through the dendrites and excitatory neurons there. So, but those don't have to do with our class at all. So this is a 3D picture of a voltage-gated sodium channel. You have this extracellular funnel, a selectivity filter, which is going to selectively, determine which ions will flow through it. Central cavity, and then an activation gate, and this is what's sensitive to that potential difference across the membrane. If that voltage changes from its natural state, it'll change the conformation down here and open or shut the channel itself. And this is like a ribbon or like a cone or column drawing of what a voltage-gated channel looks like. So you have 1, 2, 3, 4, 5 or 4 subunits, and then you have this activation gate and an inactivation gate too on a tether. And then the acetylcholine receptor causes ion influx. Sodium and calcium will flow through acetylcholine receptors. The extracellular side is where you have the binding sites for acetylcholine, and that causes a twist in all these subunits that are together, which will twist that, like this picture, twist that open. Here the ions won't fit through, but if you have acetylcholine bound, it kind of alters the conformation and it twists the whole receptor, and you get opening of the channel, and you allow the ions will flow down their chemical potential gradient. You get influx of calcium and sodium. So
is the amount of energy that's put into these reactions equivalent to the amount of energy that channels through it?
So the amount of energy that's channeling through it is all, that's all expended, preparing those gradients via those ATPase transporters.
So it'd be like
so you burn a ton of ATP. Your brain, I think, consumes more energy than anything else, and it's all transporting ions out of the cell and into the cell. So you generate that electrochemical gradient, and then when the acetylcholine binds, it causes a conformational change, and that's energy that it's putting in, because it would rather look like this when the acetylcholine's bound. It has it's lower on the free energy scale in this shape rather than this shape. These will spontaneously open too, but it's like an equilibrium, right? Just like allosterism. So you have, this would be the tight state and this would be the loose state, right? So binding of acetylcholine, you could consider acetylcholine like an allosteric activator of the channel. It fixes it in this open state. Does that make sense? So, and then this is what I was telling you, this is acetylcholine. So you got like free rotation around a lot of these. And nicotine has a fixed structure, and so does muscarinic, or not muscarin, or something like that. And they're basically opposites of each other, so they won't activate, nicotine won't activate a muscarinic receptor and vice versa. And then we have our extracellular signaling, such as the integrins that mediate cell adhesion. You have this extracellular domain that interacts with an arginine, glycine, aspartic acid containing proteins, and it'll interact with collagen, fibrinogen, fibronectin, and others. So collagen's involved with the extracellular matrix. Where do we get fibrinogen? Yeah, formation of clots, right? Same thing with fibronectin, right? And that have to do with clot formation. So if you have, let's say, I think these integrins that interact with fibrinogen are going to respond to clotting, basically, right? And they'll alter the shape there. So this triggers a cytoskeletal rearrangement and gene expression, and the newly expressed genes bind to the intracellular domain, triggering that extracellular response. And this is the example they show. So you have collagen outside, in this extracellular matrix, it'll trigger an outside signal in. So you have the inactive integrin binds to the collagen, which will trigger a signal inside via the cytoskeleton, and it'll also cause responses which will set up, polarity of cells. Like if you look at those gut cells inside your gut we looked at the other day, they're totally different from one side of the cell to the other based on which directions it's facing, right? So it's all smooth on the I forget my words. But they have like fingers sticking out on one side into the lumen of your gut, and towards your tissues they're nice and smooth basically, right? And that all has to do with reactions like this. So then you get response setting up cell polarity. So that's polarity you get from the gut proliferation, changes in cytoskeleton, and then gene expression also. So, and then you have talon is a cytoskeleton triggering an inside-out signal. So if you have a talon molecule that can bind to the integrin where it binds to the cytoskeleton and the actin, it'll cause a response how the integrins are going to interact with the collagen outside. And then the direct regulation by transcription hormones. So this is what I was talking about with the steroid hormones. They're all lipids. So they can diffuse across the cell membrane and into the nucleus where they'll interact with their nuclear receptor. Once you have a bound receptor to a steroid, it can interact with its hormone response element on the genome and cause gene transcription to occur. So hormones, because they're lipids, oftentimes they'll need a hormone binding protein to transport them around in the periphery. So other examples are this bacterial chemotaxis is controlled by enzyme coupled receptors. So for example, you have some sort of attractant, binds to its receptor, causes phosphorylation, and that's going to, phosphorylated form of the component reverses the direction of the motor, so it can change the direction of the flagella on a bacteria in response to some chemical that it senses right outside its cell. Does that make sense? So these are the signaling components present in mammals, plants, and bacteria. Basically, a lot of the things we talked about already. And you see bacteria has a lot less because they're single-celled organisms that don't need to respond to other tissues in the body, right? So plants and animals, we use structurally similar signaling molecules. So plants have molecules such as jasmonate, and that's pretty similar to prostaglandin E1 and so forth. Indole 3 acetate in plants, which looks very similar to serotonin. By the way, you know the amino acid this is made out of? Tryptophan. Yeah. Well, I guess it has hydroxytryptamine. Then you have like estradiol, very similar to these plant molecules like brasa. No steroids.
Aren't the prostaglandins involved in inflammation?
They are. Yeah. So, and you remember what blocks synthesis of those? No. You remember the enzyme cyclooxygenase?
Oh, okay, yeah.
Yeah. So plants and animals, they'll use similar signal transduction pathways, so they still have protein kinase signal transduction. Activates a MAP kinase cascade, which will involve transcription, similar to ours. We'll get toll-like receptors, and that you guys were talking about earlier. Activates autophosphorylation, phosphorylate protein kinase IRAC, which will then activate a MAP kinase cascade. And you get transcription factors such as Jun and Phos, and then immune responses to that. So those immediate early genes get turned on first. Sensory perception is also mediated through G-protein coupled receptors. So vasopressin, epinephrine, G-protein coupled receptors. Then you have light. So you have rhodopsin as a G-protein coupled receptor, which decreases cyclic GMP, activating a calcium-sodium channel. odorants work in a similar fashion, and so does taste. And I think we look at different examples here. So for rhodopsins, do you guys remember there's, what molecule does this represent? Retinal, cis-retinal, right? Light will absorb, that cis double bond will absorb light and transition to a trans double bond, get excited and it drops back down to the ground state, which will then activate GTP to exchange for GDP, which then goes over to this PDE, which activates, which basically inhibits cyclic GMP production and activates cyclic GMP breakdown, which will cause calcium to flow through the cell, basically, causing an action potential eventually in the neurons. Similar things happen in nerve responses to smell. You have an odorant binding protein, bind the odorant, or the odorant itself will just bind to the odorant receptor. You get GTP exchanging for GDP again. The activated OR catalyzes GDP to GTP exchange, and the GTP activates adenylyl cyclase, so cyclic AMP gets produced, which will then activate cyclic AMP gated cation channels, allowing calcium to enter the cells, which then calcium will inhibit. So generation of nerve impulses in response to taste is very similar again, G protein coupled reception. And I don't know how many times you want me to say GTP replaces GDP. The alpha subunit diffuses and it's going to activate adenylyl cyclase and so forth. In this case, it activates protein kinase A, which phosphorylates the potassium channel, which shuts it. Cell cycles also dependent on protein kinases. So have you heard of cyclin-dependent protein kinases, CDKs? They basically regulate the cell cycle. So in G1 to GS, you have cyclin E, or cyclin-dependent kinase 2, active. From S to G2 phases, cyclin-dependent kinase 2, cyclin A, cyclin-dependent kinase 2 is active. Cyclin B, cyclin-dependent kinase 1 is active in M phase. And misregulation of these cyclin-dependent kinases will completely throw off the cell cycle and can actually trigger cancer cells if you have misregulation. So, and CDKs are regulated by phosphorylation and proteolysis. So if you have no cyclin present, cyclin-dependent kinase is inactive. Cyclin synthesis leads to its accumulation, which will then bind to a cyclin-dependent kinase. That cyclin-dependent kinase will get phosphorylated and start phosphorylating other proteins such as phosphatases and activating them, and you get the whole cascade regulating the cell cycle after that. So growth factors will trigger transcriptional regulation of CDKs. So growth factors will cause a MAP kinase cascade. I could show you those. The MAP kinase cascade. MAP kinase will phosphorylate MAP kinase. Well, no, I think MAP kinase. Have you seen those cascades where you end up MAP kinase, MAP kinase. They all phosphorylate the other one, which eventually ends up phosphorylating Jun and Phos in the nucleus, and that'll change transcription in the nucleus, turning on cyclins and transcription factors. And then you get transition from G1 into S phase. That's how that can occur. But constitutive activation of cyclin-dependent kinases will lead to tumor growth. So, and they make good targets against cancer.
When cancer cells grow, does it activate any specific energy or any specific pathway that hinders G0 formation?
That hinders what formation?
G0.
G0? I don't know.
Deactivates apoptosis a lot of times.
Yeah. So you'll deactivate apoptosis. They have mechanisms to deactivate immune responses to the cancer. Because a lot of the cancers are due to the mutations on some of the oncogenes, which are like include the cyclins and other genes that regulate that cell cycle. So when you get mutations there, enough of them, you'll have a breakdown in the regulation of the cell cycle and you'll have uncontrolled growth. But your immune system should be able to pick a lot of those off. But if you have constantly mutating cells here, eventually your immune system is going to select for the cells that are resistant to the immune system.
And metastasis is when it can spread those oncogene
Yeah.
mutations to other cells.
Well, it doesn't spread it to other cells. It's the cells are able to spread out of the tumor.
Yeah.
Yeah, so those cells will then, and they evade the immune system because,
They go out of the tumor into the bloodstream and then.
yes.
So, yeah.
So. And then protein kinase inhibitors, they are going to use, or they are targeting cyclin-dependent kinases to block those phosphorylations, regulation of the cell cycle, to try to keep it in a non-growth phase. And then we learned about damage to macromolecules can trigger programmed cell death or apoptosis. So you have FAS ligand, FAS receptor, or tumor necrosis factor alpha. So receptor can cause these caspases to become procaspase 8 and procaspase will be activated. That'll go to the mitochondria, which will interact with cytochrome C. This will go on to activate caspases 3, 6, and 7. Cytochrome C, when it's released from the mitochondria, activates AFAP1 to form the apoptosome. And you get procaspase 9 then gets activated, the caspase 9, and the DNA will be degraded. And you get the cell membrane falls apart and you get programmed cell death that way. So. And all these caspases, they're what kind of enzymes, do you know? So all these, like procaspase 8 is inactive and then it gets altered chemically. Do you remember how the caspases are turned on and off? Yeah, they're enzymes linked, but how? They're zymogens, right? They're all proteases. And they get cleaved and that activates their protease activity and they'll cleave the caspases and activate them, and it's kind of signal transduction by activating all the proteases, right? So, and that's it for this chapter, I guess. I guess I can't start the other chapter.
Yeah, I heard on the radio, it's been a few months now, that there is, I guess, some development in cancer treatment for targeting the RAS gene.
Oh yeah? How are they doing that? You what?
I don't know if that means anything to you. You know more about that.
Yeah, I'm not, cancer wasn't my field. But Alzheimer's was, and the funny thing is there's a negative correlation between Alzheimer's and cancer.
Really? So like if you're more susceptible to Alzheimer's, you're less susceptible to cancer, and vice versa?
Possibly. The people with Alzheimer's have a less incidence of cancer, and likewise people with cancer will probably, they die earlier.
I believe that my family,
have a less-
we have a running joke in my family, we live forever, we don't get cancer, but we go crazy. So.
Yeah.
I'll be the exception in my family. Maybe I'll still live forever, but I won't.
You've already made it, you've got resistance.
Yeah.
You know what to do.
Regarding the mechanisms,
Can you
Simon,
repeat that?